neuroscience technology, I want to explain you hot topics in neurophotonics, from in vivo super resolution to deep learning microscopy. Okay, you already know my names, and you see the beautiful background. This came out in 2 years ago by the group who invented super resolution, and for that he got Nobel Prize in 2014. Okay, let me start with the multi-scale optical imaging in biomedical research. We are interested in brain and nervous system, and however, you look down what's happening at the bottom of this system is, in fact, when we are using, let's say, mouse brain is about 10 millimeter scale, and this axis is penetration depth by using light. You know, light cannot penetrate our body very well. That's why you cannot see me transparent, okay? While the y-axis is resolution. So when we want to go deeper, unfortunately, our resolution is poor. So that's what you see here. This is an example that I made this mouse who have a brain tumor growing in the brain. However, that brain tumor emits light so that I know where the tumors are growing and I can estimate the size. However, you see a poor resolution of space. This is we call macroscopic. And then we go to microscopy, which means literally microscopic resolution, okay? While for this, we can only see about 20 to 1 millimeter depth. We can't go deeper than 1 millimeter to see microscopic resolution, which you will understand later on, a multiple scattering nature of the light. In between, we call it mesoscopic, and that's literally between microscopic and mesoscopic. However, you know, I'm interested in the fine process of neuron, which you see here. It's a little tiny, but you hear a tiny neuron, a protruding called synapses or dendritic spine. The size is only about even below 1 micron. It's very tiny. So you can only see within 100 micrometer of depth, but you can see very much small scale, even down to 10 nanometer. What it means is, you know, proteins and molecules have a size of nanometer or even smaller. And now there are technologies which even we can see at this nanoscopic range. You see how big these changes are? This we call multi-scale. And we are dealing with this very interesting different dimensional scale. All right, so I'm gonna go through a few number of hot topics, which is subcellular in vivo 3D super resolution microscope. And second, and cell and tissue volumetric light shift. You know, this wasn't available even 5 years ago. People didn't know much about it, okay? And the third is more of clinical relevance, is UV or ultraviolet surface microscopy for rapid slide-free histology. Histology means a study of the tissue, usually by watching under the microscope to cut through the tissue and observe to find out if there's any pathologic processes are ongoing. And the fourth is deep learning. You guys already have heard AI and deep learning. Of course, deep learning is penetrating into any of research fields. Even microscopic image analysis will see the power, and hopefully the ultimate hurdle and ultimate restriction of my imaging microscopy is deep tissue imaging. And we are currently trying to overcome this. Okay, let me start with this cover of Science in 2018. And you see very interesting title called Resolution Revolution. I think it's very much poetic and which just is an amazing way to understand what's going on. Okay, so here you see the background and you will see the movie. So here's Eric Betze, who is one of my very interesting person, Nobel laureate for the invention of super resolution microscopy, and he's now at the UC Berkeley. So let me start with the example. Adaptive optics makes it clear. This is zebrafish inner ear. You see this is immune cell and dynamically moving. The blue part are sugar molecules, which is uptaken by these immune cells. And they're like crazy working hard. This is happening at the rear inner ear of the zebrafish. And this imaging technology allows a very fine resolution. You see, this is a blood vessel and red blood cells are passing through. You see a blinking, and here is that immune cells moving around. You see how busy they are. And there's a mitosis of endothelial cells, and immune cells are being observed, so that in real time at the exquisite resolution, now we can study these phenomena in real time. So that is possible, and we can take out the immune cell, and using MATLAB and image analysis, you can see the trajectories of the immune cells in 3 dimensions. So this is where we are at. Although this technology is still not yet commercialized, but it just takes a few years for us. If we have money, we can get this, or we can develop imaging technologies like this. All right, so what is good for this? We are talking about in vivo and complex, rapidly evolving cell physiology. You know, physiology of the cell in the real intact state is just amazing, which was unimaginable for the past previous researchers, because we provide high spatial resolution, temporal resolution, and low phototoxicity. To study this, we have to shine light, and the light sometimes cause toxicity. And we can see dynamic, not just cellular, but subcellular, which is tiny, smaller organelle aspects of the cellular process. How this is possible? Because we can now develop ultra-thin light sheet from 2D optical lattices. This sounds a little difficult, but don't worry about it. And we can shine entire field of view by shine like light sheet as a plate of light. This is the light sheet. And you can consider a CT of optical version. Hey, I have this object or cell. You just cut through this light sheet and you can see the information. And you move the light sheets up and down, and you have whole 3-dimensional aspects. So illumination of entire field. And to increase the resolution, we actually do some trick, not just light sheet, but structured illumination. That means we want to make a pattern of the light sheet which codes for smaller information when it interacts with the sample, and even hundreds planes per second. Okay, so this adaptive optics lattice light sheet microscope allows now, because we see a huge things happening at the same time. Now, you know, traditional biologists just look at one at a time. Hey, I have a hypothesis, okay? But now what happens? Many interesting things you observe simultaneously. Now it's time to hypothesis generating, because there's so interesting things are happening. So that science paper is here, and I want to share a YouTube video for this. Let's see. Can you see this?
Details at an unprecedented level.
I started to work on how.
Sorry, I'm gonna start from this.
This is the miracle of being able to see what was not being able to see before.
It's more like Star Trek, right? It's just the age of exploration again.
It's simply incredible. So we biologists like to see how things occur, and we like to see a cell, we like to see the interior of a cell.
Cells didn't evolve on a cover slip. Cells didn't evolve in isolation, right? So we want to be able to study these cells in a more natural, more physiological setting.
Eric Betzig, which is the inventor of this microscope, had 2 brilliant thoughts. One was to create a form of microscopy where you have a very thin layer of light that you scan through the sample, and that allows you to minimize the damage in the samples. And the second problem that he found a solution for is he borrowed a trick that the astronomers use today with their telescopes, which is how you deblur the perturbations you get on the atmosphere when you're looking at a star. And we have the same problem when we're looking inside a tissue.
With this microscope, for the first time, you can actually observe subcellular details at an unprecedented level.
I started to work on how cells eat for many years. All my life, I dreamed to see this happening in a live organism. Finally, we have been able to do this.
Being able to observe a neutrophil just crawling through the ear of the zebrafish, scooping up all of these food particles that we've injected, that's just mind-blowing. I actually got goosebumps in my hair. I still have goosebumps thinking about the moment when we actually first saw that. Being able to visualize that was phenomenal.
Cancer cells. So these are human cancer cells that are being implanted in the zebrafish. And you can see now the cells moving along capillaries, cells crossing the wall of the capillaries. But it's not just a blob of the cell like other people have seen in the past. This time we see the details of how the cell is sending pieces of the cell to explore their space.
We've mapped the entire eye of the zebrafish and then computationally exploded the cells such that you can look at each cell as a unit. And what you can actually observe is the changes in morphology in the context of the tissue. That's never been done before. You can look at how all of the intercellular compartments are dynamically changing, right? We can see cell mitosis, cell division, cells kind of pushing against, beating up their neighbors while they're kind of violently dividing, or at least that's the impression I get when I'm looking at the cell divisions.
Now I cannot sleep anymore. I'm up every night trying to figure out how to do the next thing.
That's like one of the highlights of working at the forefront, is because you're one of the first people to see these things. You're the first person to see this even before your boss gets to see it. So that's amazing.
Up to now, the science that we have been doing is hypothesis testing. Now we can do hypothesis generating.
If it were up to me, I would spend the next couple of years just imaging and exploring, because I feel like there's a lot of unknowns. You can actually start to ask questions that can push the frontiers in new directions.
And then we can generate our thoughts on how to go next and test. That's a big, big difference.
Okay. Let's get down to...
This is the miracle of being able to...
Okay, so I think you'll see this. And so that lattice light microscope, how to develop this, you need to understand the engineering and the basic optics, how light works. And I just want to show the core of this. You need to understand Fourier transform, because this objective lens, so-called backfocal plane, you design this, which will make the shape of the focus at the sample. To make it faster, you move it through very fast so that you make a light sheet, which allows a fast imaging. And at the end, the small size of so-called point spread function gives you a high resolution. And for this dynamic moving cells, you can shine with this light sheet very fast, and you're imaging. This is shining light, this is imaging light. Okay, I don't want to get deep into this for now, but I want to give you an idea. In the past, we can image this way, but by using so-called AO, or aberration correction. So aberration correction is developed in the astronomy, so that we can watch the stars. From physics, these knowledge is actually transformed into our microscope, because there are distortions by the optical system itself and the specimen itself, and we can correct this to give an exquisite resolution. All right, so what we got from this microscope, you see developing a spine of zebrafish. You see these neurons, And this HPF 58 hours post-fertilization, the dynamic formation of the growth of nerves in the spinal cord has been figured out. And these cancer cells, you know, I studied cancer metastasis, they have to get into the blood vessel. These are now being able to study it, not only cellular level, but you see a tiny subcellular organelles are being observed. So this is now being possible. All right, so I think I'm gonna move on to show tiny organelles. The cancer cells are trying to get into these capillaries. And this we call it as a extravasation because this metastasis killed over 90% of the people, which forms a top killer in Korea. So elderly people, you see many people die of the cancer. We still are trying to understand this process, but now it's possible to use this very high resolution dynamic microscope. So another ways of making those microscope is called volumetric light sheet microscope. And this came out in Neurophotonics in 2015. You look at this, a nice naming is a must for when you publish a good journal. So it's called swept, confocally aligned, planar excitation. It's okay, it doesn't make sense to you right now, but the point is you want to make light sheet and fast moving, and that you want to image. Okay, but I want to show the results of this. Now, this is called Drosophila larva, which is, you know, fruit fly for experimental. And now what you see here is their heart and is beating in real time. So here you can see, because it's 3D microscope, different depth, we are seeing this larva moving around. And you see, this is more of a tail part. And when you go back, and this is just a color coding of different depth. You see, this is heart beating of zebrafish larva. So that people who are interested in developmental heart formation can study this behavioral aspect. And this is now all possible, like heart beating and peristaltic waves of this larva in intact moving around. And this forms gives us an enormous amount of information. And there you need to develop a way of how to analyze this, how to quantify. And that requires more of a engineering or scientific methodology. And this is an example of mouse brain in 3D and blood vessel and so-called calcium imaging, which is so-called functional imaging of apical dendrites of layer 5 neurons. And I think I'm gonna skip this. There's a YouTube video, so I request you can go to there. And this also allows at ultra high speeds and over called 20 volume per second. What it means, you know, typically when we are talking about imaging, we talk about frame per second. And if it's a video rate, we talk about 24 frame per second. But now what we are talking is not frame, volume. You know, volume is made out of multiple orders of magnitude of frames. So this is just changing our understanding and ability to study a living phenomenon. So how to make those? Ideas are usually very simple. So this is called light sheet. As I explained light sheet, how to make sheet, you can move the single one in one direction very fast. And there are ways of making these beams, sometimes called Bessel beam, which is mathematically, you can see a little better shape than this. And this will make light sheet more efficient. Okay, so, and this is one of the microscope we are developing in our lab, so-called light sheet Theta Microscope. And what we can do with this is the left side, it's a human brain. And those who know about DAPI is a standing for nucleus, okay? So these are the nucleus of human brain. Human brains are huge, so that this is a, you see these small things, it's so big that we have to stitch them. And then this is mouse brain, smaller, but you see These are special genetically modified mice which neurons will express YFP, called yellow fluorescent protein, so that you can observe at detail. Okay, and the other is open top light sheet microscope for clinical pathology. So what it means is you have a sample from the clinic, let's say breast tumor chunks. I want to see if the tumor cells are there. And now you don't have to cut through, but just put it on top of the microscope using light sheet. You can actually see 3D. And then you just move the stage in real time. You can produce images like this. And without even time-consuming clinical pathologies, H&E stain, you can obtain very similar things. And another aspect, another new tool is the ultraviolet surface microscope, because in the clinic, people are interested in the surface of what you resect. I want to see if there's a tumor left or not. So this is traditional H&E, and UV light shining will give you a newer color so that it's more colorful. And this left is H&E stain, and this UV microscope called MUSE, okay? The goddess of the music. And you see here gives a more rich information compared to this. So that provides novel contrast. Where comes this contrast? Because we know the trick of the interaction of the light and tissue, especially the proteins inside the tissue. We can shine ultraviolet, and using some of the dyes called DAPI and rhodamine, which can excite two different kinds of the structure, so that the mixture eventually gives out this beautiful color tissue. This is an example of kidney tubule, and you see this exquisite resolution and contrast. So where comes the MUSE? Microscopy with ultraviolet surface excitation, MUSE. A nice invention of the new technique. So it's a slide free, and you can even convert into conventional H&E, hematoxylin, and eosin histology, but you give even more contrast. And this becomes very rapid histology in research. So that an example of MUSE is almost looks like H&E in the hospital, and this is the hospital. So FFPE means formalin, fixed, paraffin embedded. So typical tissue processing and watching in the clinic. And it's almost very much similar. That's the point. And it came out in Nature Biomedical Engineering recently. And the last thing I want to discuss is about deep learning microscope. What it means is now deep learning is when we provide more of a solution, proven solution, you know, machine art AI can understand this and spit out even better than human can do. So training neural network, and we want to generate fluorescence imaging, which means, hey, I want to stain the nucleus, cell body, and dendrites of the neurons. And we give the solution to the computer, and we train neural network. And then you just give an image which do not have staining, just phase microscope imaging. But then this deep learning can separate them out into different portions. Let me give you an idea. So this is original image, and we stain with the fluorescence. Then this is the true, and the second is predicted. Predicted means from machine learning, true fluorescence and predicted. And there are not much differences between. So this is an era that we train computer enough that we don't even need to label the sample at all, because the computer and the algorithm will give out this. We call it as in silico labeling, which means you don't even need to label the system. So this is the results, and the computer can, from this pure no contrast image, they can separate DAPI, the nucleus, and dendrites, and axons, which is a phenomenon. That's why it came out to Cell paper in 2018. Okay, and the last one is deep tissue microscope. I won't get deep into this, but let me give you an idea. High resolution gives a Nobel Prize. What's the last thing to left in biophotonics? We can go deeper. So there are people who study this to even go deeper into deep tissue imaging microscope. Okay, this is all, and I want to stop sharing.